Thank you, President Perko, for a very kind introduction, and I'm really grateful to Dr. De Beer for inviting me for this very special lecture to honor the memory of a passionate teacher and a geneticist is one really of the joys, and to be in this fantastic hall with all of you here from City College, I'm really grateful to be here. So today I'd like to really share with you a story, how we go from something extremely rare to learn about something very, very common. And the story I'm going to tell you is the story of Rett syndrome. And I thought I'll walk you through the sort of map of my talk before we start. We're going to talk a little bit how we study a very rare single gene disorder, and from that we can maybe learn something about the more common neuropsychiatric disorders. And we're going to learn something about the brain. When we use genetic tools, we can really manipulate the brain and learn something about it. So we're going to learn about excitation and inhibition in the brain, and what does that mean for psychiatric diseases. And lastly, in the very last third of the talk, I'm going to share with you new therapeutic ways we're trying to do to handle these terrible diseases that affect so many children, and they're really devastating. Leave them with these deficits for 70 years sometimes. So as you've heard, I was slated to be a child neurologist, and I was really determined to be a clinician seeing children with neurological diseases, until I met Ashley. You see Ashley in this picture. She's 5 years old, and Ashley was a very healthy girl. She learned how to speak, how to run, socialize, feed herself, go through a book with her hands, until about 2 years of age, when she stopped talking. Instead of using her hands, she started wringing her hands, and that's why you catch her in this picture holding her hands together, and became less social. She stopped greeting her father at the door, and with time she developed one problem after another. She lost her balance, she started having seizures, her autism became more prominent, and she had problems with breathing, tremulousness, and many other problems. And that's when I met her at 5 years of age, and I was perplexed because she started life normal. She was normal for a couple of years, and then developed these symptoms. And just about that time, a paper was published by an Austrian pediatrician, by a Swedish neurologist, describing a syndrome that was first recognized by an Austrian pediatrician named Andreas Rett, and they named the syndrome after him. And in that paper, they described 35 girls from Europe that looked exactly like Ashley. No one in the United States has seen a girl like Ashley yet, so I was the first to encounter her as a resident in child neurology. And she left an impression on me. And then a week later, I was in the clinic seeing another patient, and a girl who was 12 years old walks in wringing her hands. And I quickly recognized this must be Rett syndrome. And having seen 2 in 1 week, I figured there must be more. And that's when I decided to go through the medical records with the clinic volunteers, and we found 7 more cases. And that's how I got hooked into Rett syndrome. It was really, I would say, my first observation, that a chance occurrence that I saw another girl a week later, and I was fascinated and wrote a New England Journal article about that. And once you write an article in the New England Journal of Medicine, all the patients with the same symptoms come to you. And as a resident, I immediately had 200 patients, and I decided to study this disorder. So that's really how it all started. And in this video, I want you to see a Rett syndrome patient in action. I want you to notice how they're all very similar, like the other one. This one wrings her hands, and you'll see that she's not socializing much. You'll see her rocking. That's part of the behavior we see. And when she gets up, you'll see that her gait is very unsteady. She cannot initiate the gait. She has the strength, but she can't do it, and she'll fall backward. They pretty much have a symptom of every neurologic disease you've ever encountered. Some of you have seen people with Parkinson. Some of you have seen people with dystonia, stiffness. Some of you have seen people with tremor, with autism, with epilepsy. These poor girls have the symptom of every neurological disorder you've ever encountered in one person. So, having seen, I mentioned to you, I eventually ended up as a child neurology resident seeing hundreds of patients, and I decided I'm going to go to the lab, and they're all girls. I was convinced this must be genetic. What else would cause a disease only in girls? What else would cause a disease that's always looking the same? I figured it got to be a gene. And I was lucky that I found one of the best mentors in genetics, Arthur Huda, who was willing to take me in his lab to teach me molecular genetics. And I told him, he said, what do you want to do in my lab? I said, I want to clone the Rett syndrome gene. And I have 200 families that I've gathered DNA from. And he goes, show me the families, and I showed him these families. And if you look at these families, you'll notice the filled circle means an affected girl. That means in each family there's just one affected girl. There are no big families to trace the gene, to map the gene. And back then in the 80s and the 90s and even the early 2000s and even the late 2000s, this was not possible. Today, this is possible. But back then, finding a gene like this is like coming to New York City and trying to find Gabriella without having a map of the city and without having her address or her phone number. That's exactly how it was. I'm trying to find single base change in 3 billion base pairs where I don't know where the gene maps, where I don't know how to walk to it. So what helped me is that I made the hypothesis this must be on the X chromosome. Why did I think so? I thought it's on the X chromosome because I told you all the patients were girls. And as you know, girls have 2 X chromosomes and boys have 1 X chromosome. And in each of us, because boys have a single X chromosome, you can imagine that nature is not going to allow girls to get away with twice as much of anything, right? It couldn't be that girls will have twice as many genes on the X as the boys. What nature has done is made sure that in every cell only one of the X chromosomes is active and the other one is silent. So in all of us girls in this room, each of our cells will only express one of those X chromosomes, either the one from mom or the one from dad. And the other cell will have the other one. And it's salt and pepper, just like in this cartoon, you see that it's mixed. Whereas in a male, where there's only a single X, that single X is expressed in every cell. So because we only saw Rett syndrome in girls, I suspected it may be on the X. Because I suspected we never see boys because maybe they're so severe and maybe they die. We never see them. Whereas the girls, if half of their cells are normal, and only half of them have the mutation of the gene on the X, they will survive. So with that, I eliminated now everything. We're down to Manhattan, and I'm searching for the gene on the X chromosome, which is really the equivalent of finding a person now in Manhattan rather than the whole of New York City. And after 16 years, I was able to find it. It was truly brute force, just walking down the chromosome and simply going gene by gene. And it turns out to be a gene on the X chromosome, and it encodes a protein that has a long name, methylcytosine binding protein 2. I call it MECP2 for short, and I'm going to tell you in a little bit what it does. But at least having found it's on the X chromosome, that immediately helped us at least understand the syndrome better, why it's all in girls. Now, I just want to fast forward to tell you how long we have come. If I had the same 200 patients today, a single patient in a family, and I want to set out to find the Rett syndrome gene, I could find it in anywhere from 1 day to 5 days. So think about how far we've come. 16 years versus you can find it in a day. It's really, we're fortunate. It's really an exciting time in science, and it shows you the power of technology and the power of genetics, how far we have advanced. So I promised you to tell you what does MECP2 do. It is, in our DNA, there are 4 bases of DNA: cytosine, adenine, guanine, and thymine. And those are the bases that are sort of the alphabet of your genome. They're arranged in a way so that they encode for certain proteins and RNA that really determine how your cells function. Occasionally, some of these bases of DNA, particularly cytosine, can be modified. And here you can see it modified by adding a carbon atom and 3 hydrogens. That's called a methyl group. And when that cytosine is methylated, this is where the protein that's made by the Rett syndrome gene binds. It's called methyl cytosine binding protein. So this is a cytosine, that's the methyl, that's where it binds. And that's really at the heart of epigenetic, because your DNA is made from these bases, but each of us may have different methylation in different parts of our brain, and that's the variation in our genome. That's what we call epigenetics. So your genome is the DNA sequence, the bases. Your epigenome is how it can be modified, adding a methyl group, and then the type of proteins that bind and reads what happens when you have that methyl group. So this protein binds methylcytosine, and by binding methylcytosine, it can change the levels of genes. It can orchestrate how genes are regulated in our genome. When a brain cell has to function, whether to make too much or too little of a particular protein, that's what this protein does. So what happened after we found the gene? This is what happened. We discovered that loss of this protein in 50% of the cells, which means if a girl has a mutation, can cause almost majority of Rett syndrome cases, about 95% of them. So it's really a disorder that we call a single gene disorder. But it can sometimes give us milder symptoms. It can give cognitive deficits with anxiety, obsessive-compulsive disorder, sometimes cognitive deficits and seizures, and sometimes autism. I'm going to show you now 3 videos of 3 different girls that have mutations in this gene, and you're going to notice they don't look like the red girl that I showed you earlier on the video. They look a little bit different, and each of them has a slightly different presentation. Here is one who presented with autism. And you see her, she's not wringing her hand, she's very quiet, she's not interacting. Here's one who has a lot of spasticity, she's very unsteady, and she's shaky. And here's one who has uncontrollable movements. So there are quite a variety of the presentation in girls. As I said, a lot of them have Rett, but some can be different. And if we revisit X chromosome inactivation, where I told you in a girl, 50-50 of the cells are usually either the maternal X or the maternal X. So in a typical Rett girl, when we look into their brain, some Rett girls might die prematurely. When we looked, what we found is half of their cells have the normal gene, and the other half has the Rett syndrome mutated gene. Which means that if you have half of your cells mutated for this gene, you're going to get this terrible disorder, classic Rett syndrome. Whereas in these other girls that have much milder features that might present with just autism or cognitive deficits, what we find is that the majority of their cells are healthy and only few cells are expressing the mutant gene. And we call that favorable X chromosome inactivation, which means they started off 50-50, but somehow some of the cells with the healthy X were healthier, divided faster, and contributed more to the tissue of the embryo, and that's how you end up with much milder disease. How about the males? We ended up finding some males, and as predicted, they were very, very sick. Because remember, the males have a single X chromosome, and when that X gene on the X is not working, unfortunately, they are born ill, and they die within the first year of life. But sometimes, when a gene is missing, it's very severe. But sometimes you can have what we call very mild mutation, a mutation that barely changes the function of the protein. It changes one amino acid and barely affected. And we discover these males might present with autism or cognitive deficit, anxiety, sometimes bipolar, sometimes juvenile onset schizophrenia. So this way we learn now, we started with Rett syndrome, found the gene, now we see that the gene depending on X inactivation in girls, the severity of the mutation in boys, you can get from anywhere from severe death to mild cognitive deficits and psychiatric symptoms. So what happens if you have the gene? The gene doesn't mean therapy. to really start gaining insight about the disease, you have to really study it in animal model. Because fortunately, the girls with this disease can live a normal lifespan. They live into their 70s. It's not a degenerative disease. So the best way to do that, when a gene is conserved in different species, is to work with the mouse. The mouse is a very valuable genetic tool, and we created a mouse model for Rett syndrome, and many others did as well. And to our surprise, it reproduced all the features of the disease. You'll see that they have learning and memory deficits, all the symptoms, the tremors, including these unusual hand movements you saw in the girls. I'm going to show you a video, and you're going to see the mice reading their forepaws, just like the Rett girls. And altered sociability, and the males, just like human males, will die prematurely. So here's a healthy mouse. You can see its paws are apart, and it's trying to get out of the examiner. And now you're going to see the Rett mouse. And you can see how the Rett mouse is doing that same repetitive movement, and it's shaky, very much like the symptoms we see in the human patients. So in this particular disease, the mouse model really replicates all the features of the human. So once we've known that Rett is caused by losing the function of the gene, the first thing we thought, let's put the gene back and let's see if we can rescue the disease that way, because the disease is loss of the gene. And the first thing we did, we put the gene back in a healthy mouse. So remember, the mouse has its own copy, and now we added one more copy of the Rett syndrome gene to the mouse. So we ended up with a mouse with 2 copies. And to our surprise, we found that if you have 2 copies of this gene, it's not good. You also have a neurologic syndrome. So you basically, if you lose it, it's bad, and if you double it, it's bad. And you can see this mouse is also having a lot of abnormal movements and seizures. So it's unfortunate that it's progressive. And if we look at the features of this mouse, it starts hyperactive, but then it has, again, all the features of Rett syndrome. So this mouse was made, and we saw that doubling the protein is bad. We wondered if there are going to be human patients who have a doubling of their gene. Because all the Rett syndrome mutations is a change in the DNA sequence. You sequence it, you change it, and you don't make the gene anymore. But we wondered if there are humans with doubling of the gene, which means a segment of X chromosome is doubled, where now you have 2 copies of this gene. And we and others looked for that, and indeed we found that doubling the gene in males causes autism. It causes features almost like Rett syndrome. It causes cognitive problems, seizures, and again, premature mortality. Now I'm going to show you a video of my first duplication patients that we saw after we predicted that there would be human with this disorder. This is Brody, who came to us with autism, and I want you to watch how he's going to repetitively do things with his computer, with his little game computer. He has a lot of repetitive behavior and no language. And this was what his early presentation was autism, but with time, unfortunately, he deteriorated. And many of these boys sadly deteriorate, and most of them die prematurely from respiratory infections. Now, the females are milder. They typically present with anxiety and OCD, depression. Sometimes they may have a milder form of Rett-like syndrome. And this is, again, females, now you know it, it's the X chromosome, so they happen to have more of their cells with the healthy X rather than the cells with the duplicated X. So what we learned from this then is that the brain, our brain, is very sensitive to the level of this protein. You have to have it just right at 100%. If you have twice as much of it, you're going to get the duplication disorder. If you have 50%, you're going to have neurologic problems. If you don't have it, it causes early lethality. And we found patients, and we made mice that have tripled the levels, and those are very, very sick. So it's really important to have it right. But now that you have this, you can begin to look inside the brain and see what happens. I'm just going to show you one example here. What we found out is that if we look at neurons in our hippocampus, the hippocampus is the part of the brain that all of you are engaging today because you're listening to my talk and trying to remember something from that. So it's very important for learning and memory. And the reason it's really important is because it takes the information, stores it, and it sends it talks to the cortex, the hippocampus, there's connection with the cortex, and you store the information. And all of that happens in these little green dots. This is a neuron, and these little green dots are what we call synapses. Later in the talk, I'm going to show you a cartoon of a synapse to see what's all is inside a synapse. But these synapses are really important. That's what gets lost when we get older, and of course it gets lost much more severely in Alzheimer's. So here's a healthy neuron with healthy synapses. Here's a neuron from a mouse that doesn't have that syndrome gene, and I'm sure you can appreciate how few are the synapses. Here's a neuron from the mouse that have twice much the level of that protein, the duplication mouse. And you see there are way too many synapses here. So again, we have to have just the right number of synapses. We can't learn and retain if we have too many synapses. We just have to have it right. So with this, then, we decided using the mice is really valuable. We've learned so much from them. And here are all the features of Rett syndrome. I've shared them with you before: anxiety, abnormal stress, autism, etc., etc., etc. The question is, how can we tell which brain cells make which of these features? And the mouse is a very powerful tool, as a genetic tool to do that. So what you can do, you can engineer mice that lack the gene in only one type of cells. Here, for example, we engineered mice that lacks it in the serotonin cells. Serotonin is important for mood. Here are some that lack it in the dopamine. Dopamine cells degenerate in Parkinson's disease. And lo and behold, when we take this gene away only from dopamine cells, we get the motor problem that these patients have. And so on, so on. Cholinergic cells, they're really important for memory. When we took it away from cholinergic cells, the mouse couldn't remember anything, not even for 5 minutes. So we did all that, and what I'm just going to, instead of walk you through lots of data, I'm just going to share with you how we do this and tell you a little bit we've learned that's really new and important. What we can do, actually, we can do genetic tricks to take the gene away just from one type of neuron and keep it present in all the rest. We can also do it where it's missing everywhere and we only bring it back in the same kind of neuron. So we call that loss and replacement in one type. And in this way you can get to learn the most about the function of these neurons. And because we have such robust behaviors in the animal model, we can begin to map certain psychiatric and neurologic features to different types of neurons. Because the neurons never die when you take away the red gene. What happens to the neurons when you take away the red gene? You just partially disable them. And this tells you what happens when you partially disable different types of neurons in our brain. So the two neurons I'm going to tell you about today are really important. One type are called the excitatory neurons, and those make up 80% of our brain cells. And another type is called the inhibitory neurons. So excitatory neurons make the neurotransmitter called glutamate, and inhibitory neurons make the neurotransmitter named GABA. Some of you have taken Valium or some medications to help people put to sleep. Those potentiate the GABA activity. That's the inhibitory neurons. So those make 20% of the cells, but they're really important. The excitatory neurons are the neurons sort of that allow all the flow of information in your head. So think of the excitatory neurons as cars on a highway or a fast road, information is going. But of course, if the cars aren't regulated, eventually there'll be accidents. So you need something to stop the flow of information in time when the information is flowing. And that's what the inhibitory neurons do. So the inhibitory neurons can modulate when that information is flowing. So the first thing I'm going to tell you, these make 20% of the cells. When we took the gene out of those 20% of the cells, this is what happened. We practically reduced all the features of the syndrome, the motor problems, learning and memory problems, balance problems, and so on, including premature death. So those 20% of the cells are so important. And here you see these mice have the abnormal forepaw abnormalities. They have so many stereotyped compulsive behaviors. You see the mouse here lacking the gene in the inhibitory cell, and it pretty much constantly grooms to the point it takes the skin and the fur of its body. So it's really very abnormal and very sad. And in this slide, you'll see work by another graduate student who took it out only from the excitatory neuron, and that created motor abnormalities and anxiety-like behaviors, obesity, tremors, and premature death. So now when we look at what happens when you take it out from inhibitory neuron or excitatory neuron, you'll notice that some repetitive behavior, social problems, impaired learning and memory is much more sensitive to the inhibitory neuron. Anxiety and shakiness, tremor is more important, sorry about that, to the excitatory neurons. And of course, vital functions like premature lethality and body weight are regulated by both, and so is motor function. So this told us from these studies, and what we learned is that very slight reduction, because we measured GABA, we measured the signaling from these neurons, very slight reduction in these neurotransmitters and function leads to a multitude of neuropsychiatric phenotype. And this is really important because Red, as I told you, these features are many, many, many. Can you imagine now maybe, and we knew that decreasing GABA by 30% give you all these problems. Can you imagine maybe very subtle, maybe 5 to 10% decrease in GABA that might only present with autism or might only present with schizophrenia. So this will help us interpret genome sequencing in sporadic patients with these psychiatric diseases. And of course, it also guided us that the therapy will require that we work in both systems, because if we only work on one system, we will tip the balance and we might actually only rescue part of the symptoms. So we learned quite a bit from Rett syndrome, really looking at excitation and inhibition and the importance of that balance and the features that each can cause. And this led us to explore another disorder that I'm going to share with you that we did not anticipate, that is about a protein that works at the synapse and that really illuminated for us another class of neuropsychiatric disorders. So remember the green spots I showed you on a neuron, the multitude? I told you I'm going to show you a cartoon to show you what they really look like. This is what they actually look like. This is one of those little green spots. And for simplicity, so they can have a slide that's interpretable, I'm probably showing less than 5% of the proteins in that synapse. So it just shows you it's amazing that our brains are always functioning well, because so many things can go wrong. So in this synapse, there's these proteins called shank that are important. They're scaffold protein, and they're really important to make sure that the synapse structure looks good and the communication between the cell body and receiving information from other cells is good. And there are 3 types of shank genes in our body, SHANK1, SHANK2, and SHANK3, and each of them has been associated with different childhood disorders, such as autism, intellectual disability, and SHANK3 with the syndrome that has autism and many other features, intellectual disability, and even rare cases of schizophrenia. So we call all these diseases shankopathies because it's a pathology due to the loss of shank function. Typically, each of us have 2 copies of shank in our body because these are on autosomes, they're not on the X. And losing 1 copy of any of those can cause these problems. So I became interested in this protein because I learned that there are some patients that have a big duplication on chromosome 22, and that spans the SHANK3 gene, and this duplication can present either with Asperger, a type of autism where they have good intelligence, but they have social problems, attention deficit disorder and hyperactivity, and schizophrenia. But what I learned is that this duplication on chromosome 22 is really big, many, many genes duplicated, so we can never be sure which of those genes is causing the schizophrenia, motor problems, restlessness, and all of that. So again, using mice as a tool, we can test the hypothesis. We can ask what happens if we only add an extra copy of SHANK3? Would doubling the level of SHANK3 cause this, or is it really any one of those genes? And this is work done by a team of fellows in my lab. Cahoon made the mouse. Jimmy Holder is child neurologist, geneticist, helped him interpret the clinical implication, and Christian found the human patients that I'll tell you about, and we did all the physiology work. So we made a mouse that had an extra copy of the SHANK3 gene. So this mouse has now 50% more SHANK3. I'm going to show you videos of these mice. And none of you have ever worked with mice, and none of you are neuroscientists or behavioral geneticists, but I bet you each of you is going to know which mouse has the SHANK3 duplication. So there's going to be healthy mice in the cage and one mouse that's abnormal, and I think you'll know which one is abnormal. So you can tell there's one mouse that cannot sit still, constantly on the move, constantly going. And if you go into the mouse room, that's how I was intrigued, you hear ruckus in the cages because it's constantly moving. So when I saw this, I said, aha, having an extra copy of SHANK3 really causes hyperactivity. So now we know that's what it is. And here we measure activity in males and in females. And you see the normal, and you see that both the males and the females have increased activity. So we wanted to know what do they model, but the problem, what we discovered, is typically you prove somebody has hyperactivity is by giving them amphetamine-like drugs. You know, we give a human patient Ritalin or we give them Adderall. You give them that, and normally that would calm them down. It has a paradoxical effect in people with true hyperactivity. It calms them down. So we gave these mice these medications, and they became super hyper. Totally hyper, uncontrollably hyper. So then we knew they don't have the model of ADHD. So we wanted to know what would they model. And when you think about it, I mean, many of you have seen individuals who are hyper. They really can be either because of hyperactivity, but they also could be because of mania. So we wanted to see if this is modeling mania. And we did a lot of tests. I'm not going to show you all, but I'm going to show you one example of one test. There's a test where we hold the mouse by the tail, and typically a mouse quickly will become helpless and will stop fighting you. It will just hold still. And you measure how long they hold still, but not a manic mouse. So here's an example of a healthy mouse, and you'll see it's very still. It's almost like it's not moving. And now you can see the mouse with extra copy of SHANK3, it will not hold still. It's constantly moving, and you measure it. The healthy mice spend a lot of time not moving, but the ones with the extra SHANK are always moving. And then we measured a lot of other things. They overeat, they don't sleep when they're supposed to sleep, their circadian rhythm is disturbed, just very much like individuals with mania-like behavior. They have enhanced startled, and so on and so forth. So now we asked, now we know that 50% more of SHANK3, could there be human beings that have an extra copy of SHANK3, and what would they look like? So this is now the second time that we make a mouse model that led us back to the clinic. And so we went to the clinic, and we went into our genomic databases, and we found indeed individuals that have an extra copy on chromosome 22 of only SHANK3. And this first one, she was an 11-year-old who was diagnosed as ADHD. Because if you see a child who's hyper, it doesn't occur to you they're manic. You might diagnose them with ADHD. But she was easily overstimulated, had history of seizures, had abnormal social interactions, she overate, hyperphagia, she had kleptomania. And when they treated her with the amphetamine, she did not respond. So that was a clue to us that she really did not have ADHD. We found another patient who was a 35-year-old, the history of seizures, and he had the diagnosis of bipolar, he had mood swings, he had anger issues, and he attempted suicide. So then, from making the mouse, we came back to a clinical entity that we did not know that it existed, and now we know that there are patients with this disorder, and since then we found additional ones. But notice, both patients had seizures. We had not paid attention if our mice had seizures. So now the clinic informed us to go back and look at our mice, and we did. And you see here the EEG of healthy mice, and you can tell you don't have to be an expert that the EEG of a mouse with extra copy of SHANK3 is very abnormal, lots more spiking, lots more electric activity, and they have seizures. So now we have a mouse model, we have a human disease that matches it, they have seizure, they have mania-like behavior. The first thing we did, we tried lithium, because that's a common treatment for bipolar. They did not respond. So then we tried valproic acid, which is an FDA-approved drug, and we immediately normalized their activity. We reduced their activity closer to normal level, and their EEG, that was very abnormal before, you can see here, much more normal and no more seizures. So what we learned from these studies then, that SHANK3 overexpression causes mania-like features and seizures in both humans and mice. And I'm not going to tell you, we figured out the mechanism, why is that, and this mechanism is going to help us understand the more common bipolar disease. And we also learned that the importance of pharmacogenetic. Not every patient with a symptom can be treated with the same drug. In this particular type of disease, valproic acid is far better than lithium. It did not respond to lithium. So we learned that could be also helpful to these patients. And from this, because we've learned so much about SHANK3 function that I don't have time to tell you, we decided we have now a deeper knowledge of proteins at the synapse that could be important for the excitation, which we discovered that mania is from too much excitation at those synapses. We're now interested in studying families with bipolar disease. We're collecting any families that may have 2 or 3 individuals and sequencing their genomes. So can we interpret the genome in the biology we have just learned? So in the very last part of the talk, I want to move on now to interventions. These are tough diseases, and SHANK3 is easy because we immediately found an FDA-approved drug that can help the patients. But Rett syndrome, it's not so easy. And what I didn't tell you is that autism and intellectual disability make up almost 1% to 2% of newborn children these days. It's really a very high rate of problems. And many of patients with intellectual disability, with autism, also have intellectual disability. The sad thing is that there are over now hundreds of genes that will cause autism and intellectual disability. Believe it or not, Rett is one of the more common ones. But there's some autism forms that there may be 5 individuals where a gene is known, and there may be 50 individuals. So most genes account for a very, very small percentage of cases with autism, which means we're not going to be able to treat these kids one gene at a time. Since we cloned the gene, I've been studying Rett syndrome for 16 years. We don't have a pill yet. So imagine if you're going to study a disease where 5 kids in the world are affected. There are tens of thousands of kids affected with Rett syndrome. It's going to be challenging. So this is why we wanted to explore something really different. One of the ideas was, maybe, since all of them have intellectual disability, and I told you for you to learn, your hippocampus has to work well, maybe instead of focusing on the genes for therapy, maybe we can focus on modulating the activity of the brain network. Instead of really going into the gene, go to where the abnormality is, where the hippocampus is not working, and see if you can modulate that. And the technology we wanted to use is called deep brain stimulation. It's been pioneered by Levine-Gabriella in France. He developed it for Parkinson's disease. And today there are over 200,000 patients who've been treated by that, where they stimulate a specific part of their brain to control a very uncontrollable tremor. Tremor are really hard to control by medication. And since then it's been used for tremor and stiffness, the dystonia, a little bit perhaps of epilepsy. And it's being tried now, I'll tell you about that, for Alzheimer's. So nobody has ever tried it for learning and memory, cognitive deficit, particularly in a childhood disorder. But we knew about a trial where if you stimulate a particular part of the brain in human patients, you might enhance the function of their hippocampus. And in this trial in Alzheimer's that's still ongoing, they found that they can perhaps slow, we don't know yet for sure, but perhaps they slow the course of Alzheimer's. And we rationalized that children have much healthier brain. So maybe if we did deep brain stimulation, we can actually get a much better result. So we wanted to try that in our rat mice because they have very robust learning and memory problems, and we know the plasticity of their brain cell, which means the adaptability of a brain cell to take information and store it and take new information and so on and so forth, was lost in these mice. And for this, I collaborated with my colleague, Jean-Rong Tang, who is the head of our in vivo physiology core, and he has a very talented postdoc, Xuan Hao, who did also the work with him. And it's really neat. We do deep brain stimulation in human, and it's really neat how you do it in mice. You put very, very tiny electrodes, and you put them right where they want to stimulate, and you stimulate. And if you were to come and visit me and walk by these mice, you will not be able to tell a mouse is undergoing stimulation. They don't feel anything. We use the same paradigm that's used in human patients, so it's very good level of stimulation that's really tolerated. And I'm not going to walk you through the data, I'm just going to summarize because this was published in Nature a couple of months ago. What we discovered that deep brain stimulation in the rat mice rescued all their hippocampal functions. It rescued their ability to remember, it rescued their ability to learn and remember, it normalized the plasticity of the neurons in the hippocampus. Also, they had a marked decrease in neurogenesis, their ability to make neuron, and it normalized that. So this was quite exciting, but it's very much the beginning. What we would like to do now is determine if we can apply this to other mouse models of other types of intellectual disability. Because remember, we started so that we can maybe help a broader set of patients rather than just Rett syndrome. The other thing we have to really fine-tune is the frequency and the duration of deep brain stimulation in the Rett mouse model. I must say the neurosurgeons are extremely comfortable in applying our protocol because it's been done in humans and it's really much easier to get approval from the FDA for something like this than say a pill, because this has been tried in children and in adults for other purposes. But for us, this is only going to treat part of the symptoms for Rett syndrome. And if you talk to the parents, they'll tell you motor problems is another thing that's really, really impairing these children. So now we want to find out, we figured out where to stimulate to increase learning and memory. Now we have to figure out where do we want to stimulate to improve their motor function. We saw the girl freezing, she couldn't walk. How can we make that better? You show the constant hand wringing. Can we help stop that? So I'm just going to show you a couple of videos. This is work in progress. And the videos I'm going to show you how we can look at the neurons that are important for motor activity. So what you're going to see is a mouse on a ball. And first you're going to see a still mouse. And that mouse is hanging still on a ball. And then you're going to see some little, very rare green spots. Those green spots are brain cells. We take a piece of the skull, we can see the brain. We have engineered these mice to express a special gene that's sensitive to calcium. And whenever there is a calcium in a brain cell, the cell will turn green. Because, of course, for a brain cell to fire, it has to have high calcium in it. So whenever there is calcium, the green fluorescent protein will fluoresce, and this way you'll know the brain is active. So what you're going to see is very few cells are active when the mouse is resting on the ball. Then you're going to see the mouse running, and you're going to see a lot of green cells firing. So I want you to know you're watching brain cells firing in a healthy, active mouse. This is really the beauty of the new technologies we can use today, and what we can do then compare the mutant mice, the Rett mice, to the healthy mice and begin to map how different they are. So let's first look at a quiet mouse, and you can see occasionally there is occasional green fluorescence because the mouse is really quiet. There's a big microscope, it's a 2-photon microscope that's hanging over the open skull, peering into the brain of this mouse. Let's now look at a mouse that's active. So here's the mouse running on the ball, and I hope you can appreciate that you can see a lot more green cells firing. Unfortunately, the room was dim to be beautiful, but you can appreciate that a lot more cells are firing, so we can record, we can compute, we can tell how many, the pattern of the behavior of these neurons, how well they fire, and compare normal and disease models. And we can then draw conclusions and decide what's the source of that abnormality. So this is one example of new things we're doing. The second type of therapy I'm going to share with you is also very new. And this is to think about the duplication syndrome. You remember I told you that disease is caused by an extra copy of a gene. And this is work of Hezi Steinberg in my lab. Wonderful fellow. We asked a very simple question. If you have an extra copy of a protein, if you eliminate part of the amount of the protein, bring it back to normal, could we help it? Because if we could do that, then you can do that for the duplication syndrome, you can do that for the Shank-free duplication syndrome, and so on and so forth. So and we wanted to use a technology that we can take into human beings and really use it in the clinic. So the technology we decided to use is called antisense oligonucleotides, and the idea is we put a little pump in to infuse things into the ventricle of the brain of the mice, and hope that this will bring back the levels of MACP2 levels to normal. They're twice normal in our duplication mouse. So what are antisense oligos? Imagine that this is the mRNA. The DNA makes mRNA, and the mRNA codes for the protein. So imagine this is the MACP2 mRNA. If you add this little tiny piece of oligo, it's a tiny, tiny small piece of oligonucleotide. If you add it to match a piece on the MACP2 RNA, the minute they get together, an enzyme will see that and will immediately degrade that RNA so you don't make a protein anymore, so you can normalize the protein. So we did this test trial in our duplication mice. We allowed the mice with the duplication to grow up into adults, to develop all the symptoms of the disease, because I told you these mice model duplication in human with seizures. You've seen the video of these mice early on. And then we infused the ASO in the brain very slowly over 4 weeks. We measure MACP2 because we're going to get rid of the extra copy that we added and see that it gets back to normal. And the question we asked, after the mice are adult and have all the neurological problems that these kids have, if we now normalize the protein, can we normalize their behavior? Can we rescue them? Or is it already too late? Is it done? We can't reverse. And second, does this technology work? First thing we do is, I told you we put it in the ventricle, but we measured these ASOs and we found them in all brain region. Second, we measure if the levels goes back to normal. So here's a healthy mouse, very low level of the protein. Here's the mouse with the extra copy, high level. Here's a mouse who got the ASO. It gets to be similar to normal in 2 different brain regions shown here. Now I'm going to show you 3 examples of behaviors we do that these mice have to tell you how we quantitatively measure if there was a rescue. So, usually the examiner is blinded. They don't know if these mice got the placebo, we put just water in the pump, or got the ASO. And here's an example of activity. We measure the activity, how fast and how much the mice run. And you can tell healthy mice run very well. That's an example of a healthy mouse. The transgenic or duplication mice are not very active. You can see that in the middle. And then here's the mice after we gave them that ASO to normalize the level of the protein. They look much closer to the healthy mice. These mice have anxiety. How do you measure anxiety in a mouse? Here's an example. You put them in this plus maze, and there's a closed arm and an open arm. And the mouse hates being in the open because it feels unsafe. They like to be, hidden. You always see them running under something to hide. They don't like to be in the open. And again, the more anxious they are, the more they want to be hidden, the more daring and comfortable they are, the more they'll be in the open. So you see the healthy mice spend time in the open, but the duplication mice are very anxious, whereas after ASO treatment, now you see them venturing more, less anxious. We measure social behavior. You put a mouse in this maze, and here you have an empty cup, here you have a cup with another mouse. A mouse that has more autism-like feature doesn't really care that there is an animal here. It will go into any of these chambers equally. It doesn't pay attention to social interaction. But a healthy social mouse will spend more time here. So again, we measured that, and you can see that these mice do much better on this test. And finally, these mice have terrible seizures, and we asked, if we treat them now when they're 7 months old, really, that's old by the age of the mice. Mice live 2 years. So 7-month-old mouse is like a person maybe in their 40s. And if you treat them, and what will happen? And usually, the healthy mice have a normal EEG. The duplication mice have seizures. After this treatment, we got rid of the seizures, and they've done much better. So they don't have clinical seizures, and their EEG is normalized. So what we learned then from this reversibility studies is that if we can, in this duplication disorder, bring back the level of the protein from twice normal to normal, and in this case, we did it by this ASO therapy, we can reverse the behavior, the EEG, and all the other molecular problems I didn't share with you today. And we also tested this on cell lines from the human patients who have the duplication, and we could bring it back to normal. And what we think this has a broader application. You can imagine the SHANK3 duplication. You could use an ASO for the SHANK3 gene and hope to normalize the level. You can imagine Down syndrome, where they have a copy of a whole chromosome, and we know some of the genes on that chromosome, doubling them, gives them the risk for Alzheimer, for example. Down syndrome patients get Alzheimer at 35 or 40. If we only got rid of the extra copy of the APP, we can at least, as Down syndrome patients learn, they can stay with what they learn rather than developing Alzheimer's. So these are all examples of what can be done. And I should mention to you that ASOs have been approved for clinical trials for another disorder that's involved the muscle and the nerve. It's called spinal muscular atrophy. So we know they have a good safety profile and they could be used in human. That's why we're excited on developing this into future therapy. So now, to summarize for you everything I shared with you today, I hope I convinced you that studying a rare single gene disorder is giving us insight into causes and mechanisms for the more common neuropsychiatric diseases, and that the research using mouse model has taught us which neurons are causing which psychiatric features, and sometimes they take us back to the clinic. Lastly, I told you today the story about 2 proteins for which the brain is very sensitive to. I call this the Goldilocks principle. If you have a little too little or a little too much, it's bad for the brain. You have to get it just right. And that manipulation of parts of the circuit might alleviate some red-like features. And now, using this new ASO approach, we might translate that, hopefully, to human patients to treat the duplication disorder. And of course, the work could not have been done if not for courageous postdocs. Ruthie Amir, I would say, is the most courageous of them all. She is the one who stuck with me trying to find the Rett syndrome gene, and she did, when nobody believed there was even a gene to be found, and then many others who contributed to many studies. And of course, we were grateful to all the families who participated in the studies and the funding agencies. Thank you. Be happy to answer questions. And there are microphones if you'd like to come up to the microphone. Either the talk was terrible, nobody understood anything, or it was super clear. If you have no questions. Yes.
We figure out why it started with reverse here too. Was it just that it was half the excess of development, or?
Yes, so that would be a very excellent question. Whenever I skip a few pieces of data, there's a smart person in the audience who will make me tell you more. So the question that was asked is, why does it take 2 years to show up the features of the disorder, whereas the gene is defective from birth? And actually, X chromosome activation is set very early on. We think there are 2 reasons for the delay in the onset of the disease. The first reason, some of these sites on which the cytosine gets methylated don't happen till later on in life. Which means early on for your learning, these early milestones you learn, you don't need as much the function of this protein. You need this protein as you have more and more experiences in your storing experiences, and perhaps some of those experiences, the brain interprets them through these newly laid methylation marks on which the red protein comes and sits. So we think that's part of the reason for the delay. And the other part is probably there's a lot of excessive synapses in the first 2 years of life. And perhaps it is somehow protected that even partially functioning neurons you're still managing. And it's not until you prune a lot and you really have to really set the synaptic plasticity with the specific synapses that you start seeing features of the disease. So I think it's a combination. Yes.
This is an obvious question, so I'm just going to say one word. CRISPR.
Yes. So the question is CRISPR. What is CRISPR? CRISPR is a genome editing tool where you can combine and specifically correct a defective gene. And let's say a girl with Rett syndrome, there is a change in her DNA where a C becomes an A, so the Rett gene is no longer functioning. CRISPR would allow you to come back and make the A C again, and now you have a functioning protein. So is this a good strategy for Rett syndrome? The challenge for using CRISPR in Rett syndrome is when do you want to use it? If you want to use it in utero, then you're going to have to do prenatal testing, because this is always a new mutation. We don't have it. We don't know who's going to have Rett syndrome. This just happened, a wild mutation in an egg or a sperm. If we did pre-implantation diagnosis to every person, where you take the sperms and the eggs fertilize, select the embryos, and if you find an embryo with a mutation, you can do CRISPR on that. But to do it in utero, it hasn't been tried yet in utero. It could be done in the future. Can we do it on the brain tissue? That's a tall order. Because to do CRISPR, not only do you want to put the little piece of DNA that will go to fix it, you have to add the big enzyme that has to go and clip it. People are trying CRISPR right now in peripheral tissue, as in Duchenne muscular dystrophy, because it is easy to correct in a particular way. For Rett, many patients have 300 different mutations, so you have to design protocols, almost like discovering drugs 300 times over. 300 different mutations can cause Rett syndrome, so it's challenging for this disorder. For the duplication, CRISPR will not help. The ASO is a far better approach for the duplication because it's simple, it can be done, so we hope that that will be the case. Yes.
For the MECP2 gene, doesn't discriminate which genes it binds to, or is there a specific subset of genes with the methylated
Right.
that it binds? And the deep brain stimulation, how long does that type last? How long is it effective for?
Excellent question. So the first question, I told you the MYC2 protein binds to methylated DNA, and her question, are there few genes in our genome it binds to? Which genes it binds to? How do we know that? And we and others have been doing a lot of work on that. It turns out it binds very broadly in the genome. Wherever it finds those methylated sites, it pretty much binds. It binds really a lot of sites. Exactly how we go from binding almost so many places in the genome, thousands and thousands of places, thousands and thousands of genes, to how do we get to this clinical picture is a big mystery. But what we think it's doing by binding all these places is changing the architecture of the genome. Your genome is always wrapped, with a lot of proteins, the DNA and protein interactions. So we believe this protein helps relax and maybe compact that DNA in response to neuronal activity as you're learning something. And we believe this protein may modulate that architecture, and we've tested this idea right now.
This might be outside your purview, but I was thinking about when you're talking about people that were manic and that have different neurological disorders. Has this ever been tested with people that are incarcerated?
So that's a really an interesting question. There are a handful of genes that lead to aggression and normal, manic-like behavior and so on. And people have been talking about going, into jails and people, that had violent behavior and testing this, but that has not happened.
Sorry to go into this a little bit, but since NEP2 is known to link DNA methylation to histone modification, in your studies are you looking at changes in chromatin structure and any subsequent differential gene expression?
Yes, we've done the differential gene expression, and we found that there are many genes that are really important for brain function, that are really important to keep all your neurotransmitter at the same level, and so on and so forth, that are altered. The question we have is how, and we're now looking at the 3D architecture of the genome, and that's new technology that is being done. We're collaborating with a colleague, Reis Levine-Gabriella, who's pioneered, how to interpret architecture. We're doing that.
Why is it that alcoholic salts would for decreasing mania and would reducing the SHANK3 help that as well?
Sorry, repeat the question?
How is it that alcoholic salts work and would reducing the amount of SHANK3 that is created also help with that?
Yes. So the first question, I mentioned to you that valproate, which is an FDA-approved drug, helped decrease the mania-like behavior, and she asked, how does it do it? One way it might do it, it has its inhibition, and what we discover in mania caused by SHANK3 overexpression, that there is excessive excitation at the expense of good inhibition. We found that there are more excitatory synapses and a slight reduction in inhibitory synapses. That could be one mechanism. The second question, when we normalize the level of SHANK3, yes, we correct the features of the symptoms. And I have to answer a question that I did not address for the previous person. How frequently do you have to do the DBS, the deep brain stimulation, and how long the effect lasts? When we did it for 1 hour a day for 2 weeks, and we're able to find that 3 weeks later, if we teach the animals something, they were able to learn it, plasticity was normal for 3 weeks later. And that's what I mentioned, what's next. We're looking now as how long would that effect last to know how often we have to give this treatment.
You said that the Rett syndrome that leads to loss of MECP2 is a sporadic mutation. The textbooks tell us that it is an X-linked dominant inheritance pattern. And the reason mothers of affected daughters don't have Rett syndrome is because of skewed X activation. So could you comment, is it inherited or is it sporadic?
Yes, sure. It is 99% sporadic. Occasionally, you might get inherited. We did find a couple of families that helped us localize it, where the moms were carriers, but because of X chromosome activation, they were normal. But 99% of the cases, they are sporadic, and the gene mutation actually doesn't come from the mother, it comes from the father. It happens wildly in one of the sperms, just stochastically, it just happened. So majority of them, it is X-linked and it is dominant, but it's mostly a new mutation in every case. Rarely you're going to find occasionally familial cases in less than 1%.
Just a follow-up question. On the same token, again the textbooks tell us that the males who have the mutation either die in utero or in the first year of their birth. And so how can a male survive to pass the mutation on to a daughter?
The male does not carry the mutation. As I mentioned, it's not in the body of the male, it's only in his sperm. It's a new mutation that just happened. The male will have millions of sperms, the male has billions of cells, all of them are healthy. That male, something goes wrong, one sperm will mutate, and he will have a child with Rett syndrome. That's what we call de novo, which means it happened new. And most of autism is proving to be that new mutation that happens sporadically. We didn't know this before cloning the Rett syndrome gene, but since cloning the Rett syndrome gene, people started thinking, well, maybe autism is also sporadic. And indeed, it is a sporadic and genetic disorder. And again, majority of autism cases, the new mutation comes from the father sperm. Father is healthy. He has no mutation anywhere. Only one sperm will go wrong. I make sure everybody understands that.
You mentioned the high incidence of, I think, ADD patients. Is this higher than in the past? And if so, what do you attribute it to?
So the question you're asking has nothing to do with anything I talked about today. It really has to do, is there increased incidence of attention deficit disorders? And what is that attributable to? I think there's probably increased diagnosis of attention deficit disorder rather than, there's probably, definitely, you've got 20% of the boys in school on some medication, so I would not deny that there's increased diagnosis. I think the big question is there is truly increased incidence. There's some patients or children who really deserve the treatment, but I think somehow the art of behavior modification and really teaching the child how to sit still, teaching the child how to be attentive, and from a very young age has maybe been lost. So I think some kids are more hyper and that's not anymore tolerated. So I really think there's a behavioral component rather than to be pathological attention deficit disorder. And certainly I don't think having all these electronic in a 2-year-old hand is helping. All right. Well, thank you so much. Great audience.